Bismillahirrahmanirrahim. Today we will study the body tissues. We have four major tissues in our body. First, we have the epithelial tissue. We have the connective tissue. We have the muscle tissue and the nervous tissue. Any epithelial tissue have many characters. We will study it. First character of the epithelial tissue, it covers the surface and line the cavities. This is the surface of our body and this is the cavities. The epithelial cells also are resting on a thin basement membrane. This is the thin basement membrane. This basement membrane separates the epithelial tissue from the underlying connective tissue. The epithelial cells are avascular. They have no blood supply, but they receive their nutrition by diffusion from the underlying connective tissue vessels. There is many nerve cells which supply the epithelial cells. This is the nerve fibers which supply the epithelial cells. The epithelial cells are aggregated to each other with little intercellular substance. The fifth character for the epithelium that it develops from ectoderm, mesoderm, and endoderm. The ectoderm, this is the ectoderm, will give to us the skin and the cornea. The mesoderm will give to us renal tubules and genital tract. For the endoderm, it gives to us digestive canals and blood vessels. Also, the epithelial cells have a very important character which is continuous degeneration and regeneration. They can regenerate itself by the stem cell present inside it. Now, we classify epithelial tissue according to its site. We have covering epithelial tissue, glandular epithelial tissue, neuroepithelium. The covering epithelial tissue is classified also to simple, which is formed of one layer, and stratified, which is formed of many layers. For the simple epithelium, we have many types. We have squamous according to the shape of the cell. We have squamous, cuboidal, columnar, and pseudostratified columnar. So we have four types of simple epithelium. We have squamous, which the cells are flat in shape, cuboidal, this form of cube, columnar, these tall cells, and pseudostratified columnar. Now we will begin with the simple squamous epithelium. For any epithelial tissue, we must know the shape of the cell, the shape of the nucleus, the function of this epithelium, and the sites where the epithelium is present. Now we will talk about the simple squamous epithelium. Structure, it is formed of single layer of flattened cell. This is a flat cells with flat bulging nucleus. This is a flat bulging nucleus. This is the simple squamous epithelium. For the sites of the simple squamous epithelium, it is present in the sites which need a smooth surface, like the blood vessels and the lymphatics and the heart. Blood vessels, lymphatics, and the heart. Also in the lung alveoli, which are needed for gas exchange. Also in the serous membranes, like pericardium. This is the membrane which surround the heart. Pleura, this is the membrane which surrounds the lung, and peritoneum, this is the membrane which surrounds the abdominal organ. So the serous membrane are pericardium, pleura, and peritoneum. But this serous membrane and the epithelium which line the blood vessel, they have a special name. This name is for the blood vessel, it is called endothelium. This is the endothelium. This is the simple squamous which lines the blood vessels, endothelium. And for the simple squamous which lines the serous membranes, which is called mesothelium. So we have endothelium and mesothelium. The second type for the simple epithelium is the simple cubical epithelium. Simple cubical epithelium. So the shape of the cells is cube in shape. It is a cube shape. This cube shape has a central and rounded nucleus. Central and rounded nucleus. Where I can find this cubical epithelium, it is present in the thyroid follicles in the thyroid gland, and the kidney tubules or renal tubules. So for the simple cubical, it is cube in shape with central and rounded nucleus. It is found in the thyroid follicles and renal tubules. The third type of the simple epithelium is the simple columnar epithelium. This simple columnar epithelium, the cells are tall cells with basal and oval nuclei. So the simple columnar epithelium is formed of tall cells with basal and oval nuclei. We classify simple columnar epithelium according to modification. We have non-modified simple columnar epithelium and modified simple columnar epithelium. The non-modified simple columnar epithelium is present in the large ducts of the gland. This is the large ducts of the gland. For the modified simple columnar, we have modification for secretion, for absorption, and for pushing fluid to one direction. For the secretory epithelium or simple columnar secretory epithelium, the shape of the cell, the cytoplasm is vacuolated. It has many vacuoles due to dissolve of the mucin granules. This is a vacuolated cytoplasm due to dissolved mucin granules. It is present in the stomach. For the simple columnar absorptive epithelium, it is the same as the simple columnar secretory, but it has a microvilli. This is the microvilli, which is called the brush border. This microvilli will increase the surface area of the cells. So it is provided with microvilli. Also, it has a goblet cells. These goblet cells will secrete mucus. So we have 2 or 3 modifications. The cytoplasm is vacuolated like the secretory epithelium. The apical border has a brush border or a microvilli to increase the surface area. Also, we have goblet cells. These goblet cells will secrete mucus. It is present in the intestine. For the simple columnar ciliated, it has a cilia. This is the cilia. In the free border, this cilia will push fluids and particles in one direction. And this is very important that it move it in one direction. It's present in the lining of the fallopian tube and uterus. It is present in the fallopian tube and uterus. The fourth type is the pseudostratified epithelium. Pseudo means not true, stratified, it has many layers, so this epithelium appears to have many layers, but it is formed of one layer. So we call it pseudo, not true, pseudostratified epithelium. What is the structure of the pseudostratified epithelium? It is formed of one layer of cell, one layer only, not many layers. To say it is a stratified, a true stratified. It is a pseudo-stratified. This pseudo-stratified this layer of cells are crowded during development. During their development, they are crowded over each other. So, like these crowded people, we see that some people are tall and some are short, so they appear in different level. So our cells like that. The tall cells appear like that, and the short cells are squeezed between them. So the nuclei appeared in different levels. So they appear as a stratified or a pseudo-stratified, not true stratified, because the nuclei only are present in different levels, but the cells, all of cells are resting in the same basement membrane. Sites of pseudostratified epithelium, it has a very important 3 sites according to its modification also. We have ciliated pseudostratified columnar. This ciliated pseudostratified columnar, it has also goblet cells. It is present in the nasal sinuses, bronchi, and trachea. This is the pseudostratified. As we see, the nuclei are present in different levels. This is the goblet cells, is present in the nasal sinuses, trachea, and bronchi. So the first type is pseudostratified columnar ciliated with goblet cell. Pseudostratified columnar ciliated with goblet cells. The second type is pseudostratified with non-motile cilia. It has a cilia, but this cilia not motile like that. Is in the epididymis. This cilia also we can call it stereocilia. Stereocilia. So the first type is ciliated with goblet cells with motile cilia. Here we have non-motile cilia in the epididymis. This is the epididymis. These cilia are called, the non-motile cilia are called stereocilia. The third type is non-ciliated. It has no cilia. It is present in the vas deferens. The epididymis and the vas deferens, this is 2 sites in the male genital system. Now we will study the stratified epithelium. As you see, it is formed of many layers. We will start with the basal layer. The basal layer is resting on a basement membrane. And these stratified epitheliums are classified according to the shape of the top layer. We have stratified squamous, so the top layer is squamous. We have transitional, the top layer is cuboidal. The stratified columnar, the top layer is columnar. Now we will begin with the stratified squamous epithelium. We have 2 types. We have non-keratinized stratified squamous epithelium. It is not provided with keratin. Non-keratinized, it has no keratin. Keratinized stratified squamous epithelium. This epithelium has a keratin upon its layers. So we have non-keratinized stratified squamous epithelium and keratinized stratified squamous epithelium. We will begin with the non-keratinized stratified squamous epithelium. It has many layers, may reach about 30 layers. The basal cells of the non-keratinized stratified squamous epithelium is columnar cells. Now we study the columnar cells and we know it is formed of tall cells with basal and oval nuclei. So the cells are columnar or tall cells with basal and oval nuclei. These cells are tightly adherent to the basement membrane. These columnar cells are tightly adherent to the basement membrane to protect this epithelium from detachment. Also, these cells have a high power for regeneration, so they are responsible for regeneration of the non-keratinized epithelium. As we see here, there is a mitotic figures for the regeneration of the epithelium. The next layer is the intermediate layer, and this is a polyhedral. This is a polyhedral. It is not rounded or angulated, so we call it polyhedral. It seems like that, polyhedral with central and rounded nucleus. Polyhedral with central and rounded nucleus. The top layer, as we said before, it is fillet cells or squamous cells. So it is squamous cells with fillet nuclei. The keratinized stratified epithelium is the same. Like non-keratinized, it has 30 layers. It has basal columnar cells and a polyhedral cells and apical squamous cell, but they are covered with keratin, or it is a keratinized. So it is similar to non-keratinized and it is covered by a keratin. This is the keratin layer. Where we can find the non-keratinized and keratinized stratified squamous epithelium. The non-keratinized, it covers any wet surface in our body. Any wet surface, like the oral cavity, the esophagus, tympanic membrane, and cornea. We have oral cavity, esophagus, tympanic membrane in the ear, and cornea in the eye. For the keratinized stratified squamous epithelium, it is present in the epidermis of the skin. Now we will move to the transitional epithelium. Transitional means transition. It may be transformed to another thing. So transitional means it is transformed. How it is transformed, we will know now. Transitional epithelium is present in the urinary tract, in the pelvis of the kidney, in the ureter, in the urinary bladder. They can change its thickness according to the state of distension of the bladder, if the bladder is empty or the bladder is full. If the bladder is empty, it will be formed of 6 to 8 layers. The basement membrane is non-clear, non-wavy. This basement membrane is non-clear, non-wavy. Also, the basal columnar cells are low columnar and with basal and oval nuclei. The intermediate layer is polyhedral cells with rounded and central nuclei. So the basal cell and the intermediate layer are the same as stratified squamous epithelium. What is the difference? The difference in the top layer. The top layer is dome shape. As we see here, it is dome shape, convex in shape. The lower surface is concave. The lower surface is concave. The nuclei are rounded and maybe some cells may be pi-nucleated. Pi-nucleated has 2 nuclei. The top layer is covered by mucus which protects it from the effect of urine. The underlying layer under the top layer is flask in shape and occupies the concavity of the concave surface of the top layer. What is about the full bladder? There is gliding of the cells. The cells glide over each other to be formed, instead of 6 to 8 layers, to be formed of 2 layers only. The basal layer is cuboidal and the top layer is flat. This is the basal layer is cubical in shape and the top layer is flat. And this is due to gliding of the cells over each other. And this will be helped by the mucus between the cells. So when I'll talk about the urinary bladder, I must talk about the empty bladder and full bladder. Now, the last type of stratified epithelium is stratified columnar epithelium. It is the same as stratified squamous epithelium, but it is formed of about 20 layers of cells and the top layer is columnar in shape. It has two types, non-ciliated and ciliated. The non-ciliated present in the large ducts of the glands, and the ciliated, it is rare, present only in fetal esophagus. Now, we'll talk about the glandular epithelium. It is a epithelial cells which is modified for the function of secretion. So it is a modified epithelial cells. These epithelial cells is specialized in secretion. This is the epithelial cells which form a special function which is secretion. They classify glandular epithelium according to ducts, secretory part, and nature of secretion and mode of secretion. According to duct, we have 4 classifications. Firstly, we have according presence or absence of the duct, then branching of this duct. Then we will classify it according to shape of the secretory part, then branching of this secretory part. So we have duct and branching of the duct, then shape of the secretory part and branching of the secretory part. First part is the duct, if we have a duct or not. If we have a duct, we call it exocrine gland, like salivary gland. If we didn't have a duct, so it will be called endocrine, like thyroid gland. If it has a duct and secrete its secretion directly in the blood, so it is called mixed gland, like pancreas and liver. Then, according to branching of this duct may be branched or not. If it is branched, we call it compound gland. This is the duct. This duct may be branched, so we call it compound gland. If it is not, we call it simple gland. This duct is not branched. Then we move to the secretory part. The shape of the secretory part may be tubular, alveolar, or tubulo-alveolar. Tubulo-alveolar means it is formed of tubule and alveoli. This is dilated part, we can call it alveoli. Or tubular, it is formed only of tubule. Or alveolar, it is formed of alveoli. This is the dilated part, is called the alveoli. According to the shape of secretory part, we have tubulo-alveolar, like salivary gland. Tubular, like intestinal gland. Alveolar, like sebaceous and mammary gland. Sebaceous gland, this is a special gland present in the skin for lubrication of hair. And for the mammary gland, it is present in the female mammary gland. Then we will move to branching of this secretory part. It may be branched or simple. As the duct, we have compound and simple. So branched and simple, branched in the salivary gland, simple in the intestinal gland. From this table, we can summarize that the salivary gland present in these three compartments. So the salivary gland is compound tubulo-alveolar branched gland. For the intestinal and gastric gland, it is simple tubular gland. Simple tubular gland. For the sebaceous and mammary gland, they are alveolar gland also. Now we will focus on the secretory part. The secretory part is made of cells, epithelial cells. This secretory part may be unicellular, like goblet cells, which we said it before in the columnar epithelium and pseudostratified. This is the goblet cells and it secrete mucus, or multicellular, formed of many cells like salivary glands and pancreas. According to nature of secretion, this secretion may be mucus, which is secreted by the goblet cells. This mucus is poor in enzymes. Serous, which produce watery secretion like parotid gland and pancreas, or both mucoserous, which produce both types of secretion like submandibular and sublingual gland. May be oily, which secrete fat secretion in the sebaceous gland of the hair and skin and tarsal gland of the eye. Maybe the secretion may be waxy, which present in the external ear in the ceruminous gland of the ear. Also may be cellular, may produce cells like testes and ovary. Then we will classify the cells according to method or mode of secretion. What is meant by method or mode of secretion? This is what will happen to the cells during secretion. If the cells are not destroyed during secretion, we call it merocrine gland. These cells are not destroyed. They give their secretion by exocytosis. If the cell have apical part and this apical part is destroyed during secretion, we call it apocrine gland. This is present in the mammary gland. So this is merocrine, the cell is not destroyed during secretion. The apical part is destroyed during secretion in the mammary gland. And if the cell as a whole is destroyed, so we call it holocrine gland. This is present in the sebaceous gland of the skin. Now we will study the neuroepithelium. What is meant by neuroepithelium? This is also a modified epithelial cells for receiving of sensory stimuli, like the five senses in our body, like taste, hearing, olfactory. So this is the special type of epithelial cells that modified for receiving sensory stimuli. Structure, it is formed of three types of cells. The first cell is the sensory cell, which is tall cell and have a hair. This hair is the receptors for these cells and its free border. Their bases are connected or surrounded by free sensory nerve ending. The second type is the supporting cells, and this is the pale cell, also it is tall cell. The third type is the basal cell, and this basal cell is pyramidal. It is pyramidal and short cell present in on the basement membrane between the bases of the sensory and supporting cells. These basal cells act as a precursor for the above two types. Sites of the neuroepithelium, as we say, it is present in the five senses of us. We have taste buds for sensation of taste in the tongue, olfactory epithelium in the roof of the nose for smell, Also, organ of Corti in the inner ear. This is a 3 sites for the neuroepithelium. Now, how the epithelial cells are connected together to overcome any harmful substance outside or inside the cell. We have 3 types of connections. We have occludence junction, adherence junction, and desmosomes, finally the gap junction. The first junction is the occludence or tight junction. This junction present in the surface of the cells between the two cell membrane of two cells, which separates the intercellular space from the luminal fluid. The structure of the tight junction, it is formed of anastomosing ridges from both cell membranes. This is a ridge and this is the ridge. These ridges fuse with each other, forming a sealing strands that stitch the membranes together. What is the function of tight junction? It forms a collar around each cell immediately beneath the apical surface, blocking the passage of luminal content between the cells. It prevents the luminal content from entering between the cells in the intercellular spaces. This is a complete junction or full tight junction, prevent anything to pass inside the cell, and this is a damaged tight junction which allow any harmful or any substance to enter in the intercellular space. The second type is the adherence junction. The space between the two cell membrane is about 20 nanometer. It consists of intercellular protein. This yellow substance is the intercellular protein from the two adjacent cells. Also, we have actin filaments here and here. These actin filaments will connect these intercellular proteins. They not encircle the cell but appear as a circular spot. The macular adherence, this is a type of adherence junction. It is similar to adherence junction, but the two cell membranes are separated by 30 nanometer. Also, there is a cytoplasmic protein plaque present on both sides of the cell membrane. They are attached to tonofilaments, which are intermediate filaments. What is about the hemidesmosome? This hemidesmosome is the same, but they are not connected to adjacent cell. They connect the cells with the basal lamina, of the cells. So it is formed of half of the desmosome. This hemidesmosome is formed of half desmosome. The last junction is the gap junction. It is formed of numerous transmembranous channels or connexons. These channels permit the passage of organic ions and other small molecules from cytoplasm of one cell to another. As we said in our lecture, what is the basement membrane or basal lamina? The basement membrane, it lies under the epithelial cells. This is the basement membrane. Its function is supportive filtration, separation of epithelial cells from the underlying connective tissue. It consists of mixture of many proteins like collagen and laminin. It is like the cell membrane. It is formed of 3 layers by electron microscope. Lamina lucida, which is electron-lucent, lamina densa, which is electron-dense, and lamina reticularis, which is electron-lucent. So we have electron-lucent, in between there is an electron-dense area. Thank you.